This method can help answer key questions in the developmental biology field, such as the mechanisms of neuronal cell migration. The main advantage of this technique is that you can detect individual and collective cell migration in the long term.
After isolating 200 milliliters of bacterial culture by the alkaline lysis method, mix the two plasmids such that the final concentration of each is 4 micrograms per microliter. Incubate the fertile chicken eggs horizontally at 38 degrees Celsius in 70% relative humidity. After 2.5 days, use a 20 milliliter syringe with an 18-gauge needle to eliminate 5 milliliters of albumin from the pointed side of the egg. Seal the hole with tape. Next, use curved scissors to cut the top of the eggshell and open a hole 2 centimeters in diameter. Under a stereo microscope, check the developmental stage of the embryo. Seal this larger hole with tape. Continue the incubation at 38 degrees Celsius and 70% relative humidity until E5.5. Before electroporation, prepare 10 microliters of the mentioned plasmid DNA, colored with 0.5 microliters of fast green. Also prepare 10 milliliters of autoclaved PBS, containing 100 units per milliliter of penicillin and 100 micrograms per milliliter of streptomycin. Use a micropipette processor to prepare micropipettes made from glass capillary tubes. Cut the distal tip of the micropipette to an appropriate pore size for the amount of DNA to be injected.